Hello, third year, and welcome back after your lovely Easter break. And I hope you've all had a nice rest over the 2 week holiday and ready to get started again with Carry On Chromosome. So, what I'm hoping to do in the next few slides is take you through the inheritance section of this topic and hopefully guide you through some of the key terminology in the hope that it will give you a wee bit more confidence when you are tackling some genetics problems that we're going to set you in the near future. So first of all, we'll have a wee chat about inherited characteristics, and what they are is just characteristics that can be passed on from generation to generation. So we've got there that genetic information is passed on from generation to generation, and we've got three examples, so humans, animals and plants. And what I've done is I've just given you an example, one example of a characteristic from each of those organisms. So for humans, for instance, you can roll your tongue, or you can't roll your tongue. That is an inherited characteristic. So I can roll my tongue. So that's information that has been passed down to me from my parents, and I have passed that information on to my children. So animals, an example is fur colour. So again, that is the organisms can pass that information on through generation to generation. Plants are no different. They also receive genetic information from their parents, and an example there is leaf shape. So each organism, whether you're a human, animal or plant, has to receive two pieces of genetic information about each inherited characteristic. So I got my information for rolling my tongue. I got information from my mum and I got information from my dad that allows me to then roll my tongue. This wee slide excuse me, is just showing you inside the nucleus we've got our chromosomes, and our chromosomes are banded into genes, and each gene will be looking at a particular characteristic. And in this instance, we are looking at pea plants, and we are looking at the shape of pea plants. And you can see there that we've got a red chromosome and a blue chromosome. So the red chromosome is from the female gamete, and the blue chromosome is from the male gamete. So you can see here, that's what I mean by that for each characteristic we're looking at two pieces of information. One piece of information from your mum and one piece of information from your dad. This wee slide is just showing you the how information is passed on to get to the desired inherited characteristic. So the end result at the end of the diagram there, we can see pea shape. So that is the characteristic that we are actually looking at, whether peas are smooth or whether peas are wrinkled. Where does that information come from? If we go right the way back, it goes back to the DNA, the genetic information, the recipe for the information required to pass this information on, whether these peas are going to be smooth or wrinkled. So what we've got here are a list of the key genetic terms within a table, and what I would say to you on the outset is try your very best to try and get to grips with these terms as quickly as you can, because if you have these terms banked and you know what they mean, then they're certainly going to make the genetics problems that will set you in the near future just that wee bit easier. You can, produce a table if you like. A good idea at this point, when you've got new terms to learn and new definitions, are things like flashcards could potentially help you here. Things like Quizlet would be very good to get to grips with the key terms. So let's go through them. So alleles. That's different forms of a gene that produce different phenotypes. Now, if you're thinking, I don't even know what phenotypes or I don't even know what alleles mean, don't worry, because what we're going to do is throughout the slides is we're going to look at all of these key terms and look at them in a wee bit more depth. Things like dominant, that's an allele that produces a certain phenotype in an organism. It's always represented by a capital letter. So that's something to bank later on, you know, something that has a capital letter, it will have what we call a dominant allele. A gene, hopefully you know this term already from what you've studied previously, and a gene is a section of DNA on a chromosome that contains a coded instruction to make a specific protein. So a genotype, an individual's alleles for a particular characteristic. It is also written as letters. So again, phenotype and genotype are terms that we're going to be looking at quite soon. Heterozygous, so this describes an individual who has inherited two different alleles of the same gene from their parents. And again, don't worry, a new term, what does that mean? We'll come on to that in a second. Homozygous, this describes an individual who has inherited the same, so it's different from homozygous, so heterozygous, sorry. So homozygous means they have the same allele for the gene. Heterozygous, they've got two different alleles. For a phenotype, that is what the outward appearance of an individual's genotype is, what the organism physically looks like. So phenotype, think physical appearance. And recessive, that is an allele whose effect on a phenotype is often masked, and it is masked by the dominant allele. And a recessive allele is represented by a lowercase letter. So it might sound, oh my goodness, there is an awful lot of terms there, but what we're going to do is we're hopefully going to pick a few of these apart. Okay, in terms of just getting to grips with genetic terms, feel free, there is this wee clip that you can watch, and it kind of goes through and talks you through some of the key genetic terms anyway, so you can watch this at your leisure. So a phenotype. So a phenotype, remember what I said in the table, it's what an organism's physical appearance. So a phenotype is an organism's physical appearance which has resulted from the inherited characteristic. So if you were just to look at these two eyes that we have got here, you can see that they are distinctly different because they are a different colour. So that's what we mean by a physical appearance. It's what they look like. So the phenotype for the one that's on my left would be blue eyes, and the one that is on the right is brown eyes. So we would describe those eye colours as the phenotypes, and the different phenotypes that you can get are the different colours excuse me, of eyes. So that's what we call a phenotype. Physical appearance. So if we were to take these flowers, these snapdragon flowers, and if I asked you to state the phenotypes of these flowers, what do you think you would say? So have a good look at them. Remember, phenotypes is what they physically look like. So a rough idea of what the phenotypes would be. The one on the furthest left would be white, so because that's its colour, that's what it physically looks like. The one in the middle would be pink, and the one on the right would be red. So that is what these snapdragon flowers physically look like. So that's another example of a phenotype. Height in a human population. Again, have a look at the wee picture. Can you describe the phenotypes for these three people? So again, the one on the left would be small in height. The one in the middle, medium or average height. And the one on the right would be tall. So that's how we would describe their height, their phenotypes. Okay, so hopefully we're getting the idea of what a phenotype is. It's the physical appearance of an organism. So here is just a few other things that you can think about in terms of phenotypes. So for instance, are you able to describe human hair colour? What would be the phenotypes for that? So phenotypes for hair colour, you could have brown, blonde, ginger, black. These are all different hair colour types because we are describing what they physically look like. What you can do is you can go through human hair type, fur colour in mice, and height in pea plants and see if you could come up with what the phenotypes would be for those three other examples. So you could go away and test yourself if you feel up to it. Okay, so we're going to move on now and talk about genotype. So genotype, phenotype, they sound very, very similar, but they are very different in terms of their definitions. So a genotype is a set of genes found on the chromosome which control an inherited characteristic. Bear in mind there are two genes for each inherited characteristic, one from the male and one from the female. Okay, so I'm going to use eyes again as an example because we use that for phenotype. So we've got blue eyes and we have got brown eyes. But the genotype for that is represented by letters because we are looking at the genes for that particular characteristic. Okay. Now, different forms of the allele, different forms of a gene are called alleles. Okay, and we'll come on to alleles in a second. But just for now, the main difference between a genotype and a phenotype is a phenotype is what the organism physically looks like, and a genotype is a set of genes. It's the letters that describe that particular character, that inherited characteristic. So a genotype is always written in letters. Now you're probably wondering, why is the genotype for blue eyes? Why do we have two letters? Well, bear in mind you're going to get a letter from mum or the male or the female and one from the male. Okay, so you're getting your two parents, so one from each parent will then come together to give you your genotype. Okay, so genotypes is letters. So again, thinking about human hair colour, so if we were to take the example of brown hair, for instance, that would be the phenotype because you're describing the physical appearance of a particular inherited characteristic. But the genotype would be written in letters. So human hair colour, we could use the letter B if we wanted to use a specific letter, and you could have big B as a genotype, or you could have big B, little b, or you could have little b. that would be a description of a genotype. Because remember, the genotypes were the letters, whereas the phenotypes were how you would describe the physical appearance of an organism. Okay, and again, you can practice looking at human hair type, fur colour in mice, and height in pea plants, but not looking at their phenotypes this time, but looking at their genotypes. Okay, so we're going to move on now and talk about alleles. Now, alleles, like I said before, are different forms of the same gene. So, for instance, we're looking at colour of eyes in this example. We've got 3 different individuals, A, B, and C, and we're also describing what these individuals look like in terms of whether they are heterozygous, homozygous, or homozygous recessive. Okay, but for now, I'm just going to leave that one just now, because all I want you to think about just now is alleles are different forms of the same gene. Just try and get that into your head in terms of a definition. Because all of this, when we start looking at genetics problems, hopefully all of this will start to make a little bit more sense. So alleles, like I said, are different forms of the same gene. Alleles can be what we would class as dominant, but the alleles can also be recessive. So alleles which are dominant are always shown in the phenotype, so they always physically appear. And when we write the genotype, if an allele is dominant, it is always represented by a capital letter. So things like big T, big H, or big G. We tend not to use letters which are the same in the upper and lower case, because it's sometimes quite difficult to distinguish between dominant and recessive alleles. So we wouldn't use things like S, because S as a capital and S as a lowercase letter are the same. Another example off the top of my head would be W. So big W, wee w look very similar. So we tend to get letters that are they look different when they're in uppercase and lowercase. That's what I would encourage you to use letters which are easily distinguishable in the upper and lowercase. So alleles which are recessive will only be shown in the phenotype if there are 2 recessive alleles. So for instance, if you have an allele, two small t's or 2 small h's or 2 small g's, then you will possess the recessive, you will possess 2 recessive alleles. Okay. And this is just a sort of a wee stop point, if you like, just before we move on and talk about genetics crosses and starting to use these terms in a wee bit more detail. So you can kind of just have this as a wee stop point, make sure you know what a genotype is, make sure you know what a phenotype is, and make sure you understand the term alleles and what that means. And you can maybe try these few questions. So state the definition of a phenotype, state the definition of a genotype, a wee fill in the blanks, and a wee table just to try and consolidate your understanding of genotypes, phenotypes, and alleles. Okay, so now that we know the definitions of genotype, phenotype, and alleles, we now add in just a few more. You know, I've got my wee list there. We've already covered dominant and recessive to an extent, but we've not looked at them in a wee bit more detail. So what we're going to look at now are the terms true breeding. What does that mean? Dominant and recessive, but we're going to put them in practice, and I'm going to give you a few examples of that. Homozygous dominant, again, what does that mean? Heterozygous dominant, what does that mean? And homozygous recessive. And again, like I've said before, there is quite a lot of new terms, we get that. So just take a breather, you can pause this at any point, switch me off if I'm boring you, but just be mindful that these are the key terms that you're going to have to use when you start to look at and complete genetics crosses. So if you don't know these and are not comfortable with these terms, then you are going to find the crosses that wee bit more challenging. So take your time, get to know these definitions, be comfortable with them, practice them, and the genetics crosses, fingers crossed, will hopefully be okay. So here goes. Right, dominant or recessive. Okay, so something that is dominant, that is it describes the allele that always shows its effect, it always masks the recessive allele, and it is always written as a capital letter. So if something has a dominant allele, it's going to appear in the phenotype, and it's written as a capital letter when you write the genotype. If you're recessive, then that describes the allele that is always masked by a dominant allele. So in order for you to show a recessive phenotype, you must have two recessive alleles. And if you're recessive, it's always written as a lowercase letter. So for instance, a small t, or a small b, or a small h. So two lowercase letters for you to have that particular phenotype. If you're true breeding, then both alleles are the same. You're either two dominant alleles or you're two recessive alleles. And I've given you a few examples there of genotypes. Hopefully you've recognised the examples: big T, or wee t, or big B, or wee b, or big R, or wee R. What those are, the two alleles are the same because you've got big T, or wee t. So both alleles are the same. They're either two dominants or two recessive alleles. So we call that true breeding. So true breeding is when both alleles are the same. And they could be both dominant or they could be both recessive. So let's have a look at homozygous dominant and also homozygous recessive. So if you're homozygous, so homozygous means that you have two alleles that are the same, and if they are dominant alleles, or two capital letters, you're classed as homozygous dominant. So if you're homozygous dominant, this describes a genotype that contains two dominant alleles. For example, big T, or big B, or big R. So the alleles are two capital letters, so they're dominant, and homozygous, the two alleles are the same. Okay. Homozygous recessive. So homozygous, again, the only difference between the top term and this new term is the word dominant and recessive. So homozygous recessive describes a genotype that contains two recessive alleles. So you're going to have wee t, or wee b, or wee R. Okay, so you have got two alleles that are recessive. They are lowercase letters, because if you're recessive, you're always written in the lowercase. If you're dominant, you're always written in uppercase. However, there's always one. There's always one that just won't fit into either homozygous dominant or homozygous recessive. And that is if you're heterozygous dominant. Any ideas what you would be if you're heterozygous dominant? That's right. You would have a dominant allele and you would have a recessive allele. So you would be big T, wee t, or big B, wee b, or big R, wee r. So you have got both one dominant allele and one recessive allele. But because you have the dominant allele, the one dominant allele, that dominant allele will always appear in the phenotype. Okay, so we've got three terms there: homozygous dominant, homozygous recessive, and heterozygous dominant. Okay, right then. So this is just again a little exercise that you can try. This is you working out phenotypes from genotypes. So if I told you that brown hair is dominant to black hair, work out the phenotypes of the following. So if you have big B, hopefully you would have recognised that is not a phenotype, that is a genotype because it's written in letters. And you've got two dominant alleles. So that genotype, the top one, could be described as true breeding because you've got two dominant alleles the same. And that can also be classed as homozygous dominant. Now I have asked you to work out what the phenotype would be. So if I had big B, what would be the phenotype for hair? That's right, you would have brown hair because big B is dominant to wee b, which is black hair. So I've got two big Bs, so I'm going to have brown hair. If I have big B, little b, so I am not true breeding anymore. I am heterozygous dominant because I've got one dominant allele and one recessive allele. So I would be classed as heterozygous dominant. And now I'm asking you for my phenotype. So I would have brown hair again. Why? Because you would think, well, hang on a minute, you've also got an allele for black hair. But remember, the dominant allele always masks the recessive allele. So that would be big B would mask wee b. So I would have black hair there. Brown hair. Oh. So I would have brown hair for the middle one. Going on to the last one, small b. So I'm again back to being true breeding because I've got two alleles that are the same. I'm also homozygous recessive because I am small b. But again, I'm asking you my phenotype. So I don't have any dominant alleles. So that's correct. I would have black hair. Okay. So that is you working out your phenotypes from genotypes. But you can also flip that and you can work out genotypes from phenotypes. So if I said to you that tall plants is dominant to dwarf plants, so big T is dominant to wee t, work out all the possible genotypes for tall and dwarf plants. Now, how I would do this, first of all, is I know if I'm a dwarf, I've been told that I am recessive from the question because I've got a small t. So the dwarf plants must be small t, because they must have 2 recessive alleles for them to be a dwarf. If you are tall, there's 2 different possible genotypes you could be. You could be big T, or you could be big T, small t. Okay? And again, another wee stop point, another wee breather, because what we've done there is we've actually applied what we know about genotypes and phenotypes, and looked again at alleles, dominant, recessive, heterozygous, dominant, etc. And you might want to just stop for a bit, watch a few wee clips. There is a 4-minute inheritance clip, which is quite good. And we've got a wee summary of inheritance that is a wee bit longer, and that's 14 minutes. So again, these are just genetics clips that you can watch at your leisure. So what we're going to do now is we are going to use all our terms, all our terminology, and we are hopefully going to work our way through some genetics crosses. And we've got a wee picture of the main dude. This is Gregor Mendel. He is the godfather of genetics, and he studied the inheritance of different characteristics in pea plants. So he found that when he mated a true breeding tall plant with a true breeding dwarf plant, all the plants in the next generation were tall. And you've got to think to yourself, hang on a minute, there are tall ones and there are dwarf ones. Surely to goodness you would get half tall, half dwarf. But that was not the case. And what we're going to do now is we're going to look at a genetics cross that deals with what Mendel actually looked at genetically, and we're going to carry out a genetic cross. And what I would say to you here is I'm going to take you through step by step, looking at the different ways that you can present a genetic cross and start to use the terminology that we have covered in the last few slides. Okay, so it's really important now that you understand all the key terms like true breeding, homozygous recessive, homozygous dominant, heterozygous dominant, all of those key terms, alleles, genotype, phenotype, etc. So what we have here is a cross between, like I said, Mendel was quite happy playing with his peas, and he found that when he crossed a tall pea plant with a true breeding dwarf pea plant, this is how he presented his results as a genetics cross. Okay, so what we've got is, I'm just going to get my wee pen here, okay, and my wee highlighter. Hang on a minute. So we have got the phenotypes of the parents. So here are the phenotypes of the parents. We know that they are tall and they are dwarf. Okay, how do I know that? Look at the question. Okay, so a cross between a true breeding tall and a true breeding dwarf plant. We then look at the phenotypes. That's what the plants physically look like. For genotypes of parents, so what we've got is we are told in the question that we have got a tall plant and it is true breeding. So it must have the genotype big T, because it's true breeding. It can't be big T, wee t, because it's not. Big T, wee t is heterozygous dominant, so it's not true breeding. Okay, so look for those wee clues in the questions when you do them. Is it true breeding? Is it not true breeding? Also, we have got your dwarf plant, which is big T, small t. We then have a look at our gametes. Okay, so basically what we need to do is we just have to split that in two, and we're going to split this in two too. Here we go. And half of our gametes are going to have big T, the other half are going to have big T. The same with the dwarf, half our gametes are going to be small t, half our gametes are going to be small t. And what we do is we then form what's called a Punnett square. A Punnett square is just that's the square there. I'm just going to draw around it. There's your Punnett square. Okay. And you've maybe done a wee bit of this when you've maybe done a wee bit of genetics in second year, so you might recognise the wee Punnett square. And all we've done here, and it doesn't matter where, so your big T, so that's them up there, and your wee t, this is going to look like a big jigsaw puzzle, this. Wee. They're just there. Alright, and what you do is you then cross them over. So big T and little t. So when you put the two of them together, you come up with big T, little t. And some people, put small t first, small t, big T. That's okay. You're not going to, lose any marks in a test if you do that the wrong way around. But we would like you, as a sort of teaching point, the capital letter to come first before the lowercase letter. So we would put big T first and then the small t. And then we'd just do exactly the same for the other three. So big T, wee t, big T, wee t, and big T, wee t. Okay, so the F1, that is the generation, we call that the babies that these parents produce. So the first filial generation, that's what the F1 stands for. And we're going to look at just their genotypes. So remember, the genotypes are it's written in letters. It's the set of genes possessed by these organisms. And all of them, the genotypes, are going to be big T, small t. The phenotypes, so that's what they physically look like. They are all tall. Why do we know they're all tall? Because they have that elusive dominant allele. Okay, so they have this big T. Okay, so they are all tall. They would all be classed as heterozygous though. So they are not true breeding, they are heterozygous. Okay, heterozygous dominant, I should say. Okay, so happy with that? So what we could do now is we could take that generation and we could do a cross between two heterozygous tall pea plants and find out what their genotypes and phenotypes of their organisms would be. So again, setting it out as before, phenotypes of parents, we've got a tall one and we've got a dwarf one. But it's not big T this time because we're actually told in the question that it's a heterozygous tall pea plant. So that goes in here. And hold on a minute. Pause. Okay, now I don't know if you've spotted the error, but we are just going to, there we go, we're going to score this out because that should actually be, let me try and write it in, that should be tall, because we are told it is a cross between two heterozygous tall pea plants, okay? So there is a wee typo here, so that should not be dwarf, it should be two tall heterozygous pea plants. The genotypes are okay because we've got big T, wee t, and we're told they are two heterozygous tall pea plants, and that, oh, that is big T, wee t, sorry. Okay, gametes, again, what we've done before, we just halved that. Way, there we go. And again, done our Punnett square. Okay, so we've got the big T, wee t. So again, just big T and wee t, and big T and wee t, and cross them over. Okay, so for the F1 genotypes, we have got one, which is big T, which is there. So that's that one. And we have got two that are heterozygous, dominant, so that's there. And we have got one which is a dwarf. Okay, so for the F1 phenotypes, we have got a tall one, and another two tall ones, and one dwarf. So the ratio there would be 3 tall pea plants to 1 dwarf. Okay, that makes sense, hopefully. And what we've got here is a cross between a heterozygous tall pea plant and a dwarf pea plant. Okay, so again, phenotypes of parents. So always start with the question, what's it telling us? So we've got a tall one and we've got a dwarf one. We've told that the tall one is heterozygous, so big T, wee t, and the pea plant must be homozygous recessive, so wee t, and the gametes, again, like what we've done before, just split them in two. Okay, Punnett square, and again, just pop in your Punnett square down here. So there we go, big T, oh, that should be round here, way, and wee t, there we go, and wee t. It just looks like a big maze, this. It's just, I'm just trying to show you where the gametes go. Okay, and again, crossing it over, big T, little t, little t, big T, little t, and little t. So for the F1 genotypes, you've got two big T's, and you've got two small t's. So for the phenotypes, two talls to two dwarfs. Now the ratio, you can say 2 to 2, okay, because that's what we have, is 2 talls to 2 dwarfs, but we'll like to take that down to the nearest whole number the nearest whole number. So we talk about a 1-to-1 ratio. So 1 tall to 1 dwarf. Okay, got that one? Fingers crossed. Now, again, Mendel, he really loved looking at pea plants so much that it wasn't just whether they were tall or dwarf. He also looked at their flower colour as well. And again, that's just looking at a different characteristic. We're not looking at the height of the plants, but we are looking at their colour. And again, he found that when he bred red flowered plants with white flowered plants, all the offspring had red flowers. And again, why was that? Okay, there, you can look into that if you like, but that was, again, it was just a genetics cross that we'll maybe get you to do later on. Okay, so what we're going to do is we're just going to again move away from Mendel and his peas, and we're going to have a wee look at some gerbil genetics. And what we've got here are some nice wee gerbils. Okay, we've got mummy and daddy gerbil, and you know what gerbils do in lockdown, they just like to move about their cage and maybe have a wee bit of fun. And mummy and daddy gerbil have produced these 4 beautiful brown gerbils. So what we've got is Frank and Sandra, and we've got Big Frank here, okay, rocking the brown fur. Whoa, there we go. And here's Sandra, looking absolutely stunning in her white fur. And what they have produced is 4 beautiful brown fur gerbils. So just by looking at that slide, hopefully you would be able to tell me what fur is dominant. Correct, it's Big Frank's. Okay, Big Frank with his brown fur, it has the dominant characteristic. Why do we know that? Because the offspring are all brown. Okay, so what we're going to do is we're going to look at a wee cross to show this. So where's Big Frank? Oh, there he is. Hello, Big Frank. How are you doing? Okay, and we've got beautiful Sandra. And what we've got here is I've just taken you through, again, just another genetics cross to prove that last slide, that all of the offspring, when Frank and Sandra mate together, they produce little brown furred babies. So we've got Frank, brown fur, so that's the phenotype. Sandra, phenotype, white fur. Parents' genotypes, okay. We have got Big B, and we have got Sandra, who is wee B. Now, Sandra is very easy to understand that she must be wee B, because we know that she is recessive just by looking at her fur, or looking at her babies. So they were all brown. There was no white ones in the offspring that they produced. So she must be recessive. So she must be wee B for fur type. And for Frank, he could be, you could debate, well, he could be Big B, wee B. And you're right, he could be. However, we know from their offspring that all of the children have brown fur. So what we would do is we would do genetics crosses to prove that. So the gametes for Frank, all his sperm would have, or half of them would be Big B, the other half the other Big B. And for Sandra, wee B would be two wee bees. So both of these gerbils are true breeding. Again, we would do a wee Punnett square. So again, here's our wee square here of our gerbil genetics, and the F1 genotypes, all of them are big B, wee b. And the key thing here is to look at the phenotypes, because I did tell you, or I did show you in the last slide, that they all had brown fur. And we have proved that through a genetics cross that they all have brown fur. Okay, now the story doesn't end there, because Big Frank likes to pass on his brown fur, and his son, Big Frank Jr., okay, mates with Francesca. Okay, so we've got Big Frank Jr. and Francesca. Okay, and you would think two brown gerbils. We've got a dominant one there because Big Frank being Big Frank. You know, surely to goodness, all of the gerbils are going to be brown. Now, where are they? Let's have a wee look. Okay. She says, here we go. Okay, so here is Big Frank Jr. Jr. Another Big Frank Jr. Jr. Big Frank Jr. Jr. But hang on a minute. There is a wee white one. And again, what we can do is we can produce a wee genetic cross to prove this. Okay, so how come two brown furred gerbils produce a mix of brown fur and white fur gerbils? So why is this the case? So here is Big Frank Jr. Here he comes. Hello, Big Frank Jr. And there is Francesca. And again, we could just going through the genetics cross. So we've got brown fur for phenotype, brown fur for phenotype, parental genotypes. So they are big B, small b, big B, small b. And again, you're questioning, well, why are they not big B, big B? But again, we have to look at the offspring. We have to look at the babies that they produce. And when you cross two heterozygotes, or two heterozygous individuals, they are going to produce a mix of brown fur and white fur offspring. So we've got the gametes. So Big Frank, half of his sperm is going to be big B, half is going to be wee b. And Francesca, half of the eggs are going to be big B, half of the eggs are going to be wee b. And again, we do a wee Punnett square. And we have got our Punnett square here. All right, so the Punnett square, if we just do that. And we have got F1 generation genotypes. So we have got one big B, big B. So that's it there. There we have it. We've got two big B wee Bs. So the one, and there's the other one, two, and we have got wee b, wee b. Okay, one wee b, wee b. So in terms, that is what we call the F1 generation genotypes. Again, it's letters. And the phenotypes, what they are physically going to look like. And we have got three browns because we've got two big B wee Bs and one big B big B. So three of them are going to have brown fur and one of them is going to have white fur. Okay, so we get a 3-to-1 ratio. So what we've done so far between the pea plants and the gerbils is, on paper, what we have done is we have predicted what the offspring could potentially look like in terms of their phenotypes, but also tried to predict their genotypes as well. However, what you observe in practice is not necessarily what you can predict on paper. So there is a difference between observed and predicted results. And why is that? Because fertilization, whether you're a plant or an animal, relies on an element of chance, and it is a completely random process. So you do get different results observed versus predicted because fertilization is a random process. Okay. A wee exercise that you can do to prove that is you can get a coin, and you can, if you look at your coin, you've got a heads and a tails side, and heads on both sides, or if you get two coins, sorry, and you toss them both at the same time, and if both your coins land both heads up, you would be big H, big H. If you have tails on both sides when you toss them, you get wee h, wee h. But if you get a wee mix, a heads and a tail side, you can be big H, wee h. Okay, you can do this. This is just a wee exercise that I would have normally done with you in class in terms of looking at ratios and trying to look at observed versus predicted, dominant versus recessive alleles, and work out your dominant to recessive alleles. Bearing in mind that I'm telling you that heads is a dominant characteristic, that you can do it differently if you want. You could maybe make your tails dominant if you so wish. Okay, but that's just a wee exercise to prove just this element of chance when you throw coins in the air or toss coins in the air. It is an element of chance which side it lands on. Okay. And that leads us finally to family trees, which are called pedigree charts. And a pedigree chart is a type of family tree diagram. It shows the inheritance of a characteristic through several generations. So if we were to take something that you might be very familiar with, the Simpsons family tree, you've got Bart and Lisa and Maggie at the bottom, and you've got Homer and Marge being their parents. So if you take, there is your offspring or your children, and here are their parents. So Homer and Marge. Okay, now some of you might be great Simpsons fans. I must say I've watched it a few times, so I have watched this a couple of times, and so you can see Homer and Marge are Bart, Lisa and Maggie's parents, but you could also see their grandparents, Bart, Lisa and Maggie's grandparents being Abraham and Mona up there. Okay, and on Marge's side, you can see Clancy and Jackie being her parents, and you can see Patty and Selma up there being Marge's sisters. Okay, so what we're going to do is we're just going to be looking at family trees and what they can show us in terms of characteristics. So what we've got here, it just looks like lots of square and round circles, black and white, but you're often, to read a family tree, you're often given a key. And going way back to the very beginning when I was talking about being able to roll my tongue, I am female, able to roll tongue, so I would be a circle in this particular family tree. Okay, so if we look down here, here are the children, if you like, okay, which there are 4, 1, 2, 3, 4, and from the key you are able to tell whether you have male or female children and whether they are able to roll or not roll their tongue. Okay, so being able to roll your tongue, and you're female, so that's you here. If you are female, not able to roll your tongue, that's you here. Okay, and if you're male, able to roll your tongue, that's you here. And if you're male, not able to roll your tongue, that's you here. Okay, so you've got 4 children, and their parents, if we follow this up, so if we go up here and along, that's the parents here. So there is the dad, and there is the mum. Okay, so it's really vital when you are reading family trees is just following the chart. It's following the flow diagram and being able to pick out information about you know, how many children do parents A, B and C have, or how many people are able to roll their tongue in this generation. It's wee things like that. So it's being able to identify characteristics within a family tree diagram. Okay, again, this is just another example of family trees, again looking at, and this could be any characteristic, whether you're affected or unaffected. And again, it's just trying to identify on how family trees work. Okay, so again, we've got a set of, I'm just going to pick at random, here's a set of children here. Okay, so we've got 1, 2 females, so female and female. Oh, and we've got male and male, and again, that's their parents there. Okay, so again, family trees, just being able to identify what characteristics are within a family tree and being able to identify a whole variety of things from a family tree diagram. Okay, now hopefully throughout this, I know it's been quite long, but like I said, you can stop and start at any point, and hopefully this will give you sort of the tools in terms of looking at genetics crosses and the key vocab and terminology for this section to then go away and attempt your genetics crosses. All that I would say for this section, in particular the inheritance section, is practice, 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 practice. The more you practice, the better you're going to get at these different questions. Okay, again, if you've got any questions, please just get in touch with your teacher on Teams, and we'll do our best to try and support you. Okay, good luck with your genetics questions.